So as you've just heard, neurological disorders affect 1 in 5 Kiwis. And about 1 in 20 individuals will have a seizure at some stage in their life, and about 1 in 200 actually have epilepsy. And it's because of that that the World Health Organization has decreed a few years ago that epilepsy is the most common serious brain disorder worldwide. It has no age, racial, social class, national, or geographic boundaries. So here's a figure of a brain, and it's sitting on the background of EEG printout. And so what's happening in your brain right now as you listen to me is your cortical neurons are sending electrical information from one cell to the other. And that's all normal, and that's the way we want things to happen. Unfortunately, when you have an epileptic seizure, those same cortical neurons send an abnormal, very intense electrical signal to other areas of the brain, and that results in the individual either having an unusual sensation or having an unusual movement that they don't want to have, and that's what we call an epileptic seizure. Epilepsy is a group of disorders which are characterized by the tendency to have recurrent epileptic seizures. And people tend to think of epilepsy as a disease or a disorder, but just like you can see in this picture, so this is a picture of fruit, but it doesn't take a lot of observation to discern that actually there are a lot of different fruit on that paper, and each of those fruit are very different from each other. And you can define one of those fruits, so here we have an orange, by the way it looks, its shape, its taste, and that helps us to identify the different types of fruit. So epilepsy is the same. It is a group of different diseases, and these diseases are called epilepsy syndromes. And the way that we define epilepsy syndromes is it's based on the age of onset of an individual's seizures, the different types of seizures that they have in their lifetime, what's happening with their development. So in early childhood development with walking and talking, and later on in terms of cognitive function as adults. What we find on their examination, particularly their neurological examination, what comorbidities they may have. So what we mean by comorbidities is other neurological signs or symptoms that they may have. So intellectual disability, autism, attention deficit disorder. And then finally, what we see on their MRI and their EEG. So each of these individual epilepsy syndromes are defined based on characteristics in each of those boxes. So there are a lot of different epilepsy syndromes, and here is just the name of a few of them. And some of these epilepsy syndromes can be quite bad. So these blue and purple ones on this side, and some of them can be quite good. And talking about good and bad epilepsies and making a dichotomy like that is a bit artificial, well, very artificial, because epilepsy severity ranges from very good to very, very severe. But for the next part of my talk, I'm just going to sort of dichotomize them into good and bad. So the good epilepsy, so most people who have epilepsy have what we would consider a good epilepsy. And that's because they're normal people who have normal lives and function in a normal way. They have jobs, they have families, and their seizures are generally easy to treat with the anti-seizure medications that we have. And in childhood epilepsies, a lot of the children will actually outgrow their epilepsy. However, even in the people with the good epilepsies, you're more likely to have learning difficulties as a child than if you don't have epilepsy. You're more likely to have behavioral disorders such as attention deficit disorder, more likely to have mental health disorders such as depression or anxiety. And in about 35% of people, even though we try all the different anti-seizure medications available, we can't stop their seizures. And most of these individuals have got epilepsy lifelong. And then there's this awful thing called SUDEP, sudden unexplained death in epilepsy patients, which happens to even the people who have the good epilepsies. And they may only be having a few seizures a month, and yet they are found dead in the morning, and nobody's not quite sure why. The other thing is that there's a lot of social implications about having epilepsy. There's still social stigma associated with this. People can be discriminated and have reduced social interaction. They're less likely to be employed. And overall, that can result in a decreased quality of life. So even though they're good epilepsies, in some individuals, those good epilepsies still has a significant impact on their quality of life. So that's the good epilepsies. So now let's look at the bad epilepsies. So they are severely catastrophic in many cases. So the most severe type of epilepsy that we see are the developmental and epileptic encephalopathies. And this is a situation where you have a young child usually, and somewhere in the first 5 years of life, generally, they start having seizures, often frequent seizures. And in between the seizures, the electrical activity in their brain is very abnormal. And it's a combination of the seizures and the electrical activity, which means that their brain, which at this stage should be developing normally, is negatively impacted by these seizures and these abnormal electrical discharges. And those seizures and EEG discharges result in significant impact on their development. So they end up with developmental slowing and or regression. So these are, as I said, these are usually present in childhood. These children often end up with intellectual disability and autism, and it's typically difficult to treat the seizures in these bad epilepsy syndromes. And unfortunately, about a quarter of these kids don't survive until 20 years of age. So our Epilepsy Research Group at the University of Otago, our main aim of all of our research is to try to improve the quality of life for children with epilepsy and their families. And I'm really lucky to work with a fantastic group of people. So we have research assistants, PhD students, some fantastic pediatric neurologists that help us with our work, a bioinformatic postdoc, and Professor James Stanley, who's actually in the public health department, who helps us with our statistics and epidemiological projects. And I'm also really lucky that I have really close links with some exceptional researchers around the world. Science and research is a team sport. I tell everybody, you can't do anything much on your own, or you can do so much more when you collaborate with other people. So I have strong ties with the University of Melbourne, with Professor Ingrid Scheffer and Sam Berkovic, Michael Hildebrand, and at the WeHi with Melanie Barlow, and with Heather Method at St. Jude's Children's Hospital. And some of the research I'm going to present later on is with Alan Bayet and Ricky Muller from the University of Southern Denmark. So our Epilepsy Research Group at the University of Otago has our fingers in a lot of different research pies, I suppose. We are doing research on epidemiology, epilepsy in Māori, gene discovery, identifying epilepsy syndromes in the genetic epilepsies, and distinguishing what the genetic epilepsies, how they present and how they progress, the natural history of them. And then we're also involved in some drug trials. And I'm not going to be able to talk about all of this today. What I'm going to talk about is some of the first four aspects of this, and I'm going to relate particularly to some publications that we had in the last year. So first of all, let's start by talking about New Zealand epidemiological projects that we're running. So I've got two PhD students that are involved in these, Gemma Poke and Shema Ali, and I'm just going to talk about Shema's project because she's just had that published in Neurology. And so her project was looking at the epidemiology of treated epilepsy in New Zealand, a focus on ethnicity. And so what she did, so our aim was to try. There are a lot of epidemiological studies in children around the world, but there was nothing that had any data on New Zealand. And of course, New Zealand children are different from children in other places in the world for lots of different reasons. So it's really important for health resource allocation and understanding how best to help New Zealand children, that we understand what the burden of epilepsy is in New Zealand. So what she did was she used the New Zealand Pharmaceuticals database, and that is a database that keeps record of all drugs that are dispensed in New Zealand. And she got the data from all children under the age of 18 in the Wellington and Auckland and Northland region. So that's 48% of New Zealand children, and who were between 0 and 18 years of age, who had been given a prescription for or been dispensed an anti-seizure medication. Now you might think, okay, well, that sounds pretty easy. You just, pharmaceutical database shows you all the numbers and then you just count them up and divide it by the population. Wow. But unfortunately, it would be nice if it was that easy, but unfortunately, anti-seizure medications are not just prescribed for epilepsy. So they are also prescribed for headaches, for mood disorders, for pain, and for movement disorders. And so what Seamus search of the pharmaceutical database identified 2,700 children who had been prescribed or dispensed an anti-seizure medication in that year. And what she did was she went through every single one of their notes to find out why they were prescribed anti-seizure medicine. So it did take quite a bit of time. And what she found was that in New Zealand, there are 3.4 children per 1,000 children that have treated epilepsy. So that's 1 in about 280, or roughly about 4,500 children in New Zealand are treated for epilepsy. And this is a graph from her paper, and it's talking about the incidence of treated epilepsy. So you have the years across the bottom and the incidence on the side. And so what this shows is a number of people that presented with epilepsy in that year of life. And so what you can see is the biggest time you're likely to present with epilepsy is in that first year. And then through childhood, it's roughly the same incidence, and it starts to pick up a little bit again in adolescence. She looked to see whether or not you are more likely to have epilepsy depending on where your socioeconomic status was. So let me just talk you through these graphs here. So this is the rate ratio of epilepsy, and she used the NZ-DEP information or quintiles. And so how that works is all of the areas in New Zealand are divided up into social groups, and NZ-DEP quintiles 1 to 5. And so if you are living in a rich area, that you're in your NZ-DEP index is 1, and if you are in a poor area, it's 5. So you're most deprived up in this area. And so this graph is showing the risk or the likelihood of developing epilepsy compared to the wealthiest people. And so what you can see here is that if you are in the poorest, children in the poorest areas are 1.5 times more likely to have epilepsy or to be treated with epilepsy than the children in the wealthier areas. And because socioeconomic status and ethnicity are associated, she then adjusted for ethnicity. So what that means is just took that sort of out of the equation, and it didn't make much difference. So basically it is specifically about your socioeconomic status, which increases that risk of developing epilepsy. She then looked just at ethnicity. And so this is a risk ratio. Sorry, I don't know why that keeps flicking back and forth. So this is a risk ratio looking compared to European, so Pacific, Māori, and Asian children compared to European. And what you can see, what this graph shows you, is that there wasn't much difference in the prevalence of epilepsy between Māori, Pacific, and European children. However, Asian children were less likely to develop epilepsy. But again, because socioeconomic status and ethnicity quite well correlated, she then took the socioeconomic status confounding factor out of the picture. And when you remove socioeconomic status, actually, Māori children and Pacific children were less likely to have epilepsy than European children. So now I'm going to move on to talk more specifically about our Epilepsy and Māori Children project. And this basically led on from Seamus' project, and this is being undertaken by Nairi Keenan, who is a paediatric neurology advanced trainee and a Māori physician. And this is the project that was funded by the Neurological Foundation, and we're very grateful for that. So in addition, what we know is that Māori with epilepsy are, even though our Seamus project showed that they're not more likely to have treated epilepsy, we know that they're more likely to die of their epilepsy. They're more likely to present to the ED and be admitted because of their epilepsy. So they seem to have a higher burden of epilepsy, and this has been done from previous research. So what Nairi's doing is looking to see why that might be. So she's been comparing Māori children with epilepsy to non-Māori children with epilepsy, and she hasn't finished yet. Well, she has finished getting the data, but she hasn't finished analyzing the data. But what she has done some preliminary work from the Wellington region, and what she's found is that the the Māori children and non-Māori children have similar age of onset, they have similar types of epilepsy, and they have a similar severity of epilepsy. But there is a significant difference between whether or not their epilepsy has been caused by something that could be preventable. So, and when we're talking about preventable causes of epilepsy, we're talking about birth asphyxia, we're talking about congenital infections, infections of the brain afterwards, or trauma. So her project is ongoing. She has still to analyze the Auckland and Northern, put that data together with the Wellington region, and also to look at the difference between the comorbidities between the children, and really importantly, which may have explain why the burden of epilepsy seems to be higher in Māori than non-Māori, look at the management to see whether it's similar between the two, between ethnic groups. So the majority of the majority of our research in at the Epilepsy Research Group has historically been gene discovery or epilepsy genetic research. So just to, and so epilepsy can be caused by a lot of different things. It can be caused by preventable causes that I just talked about. It can have structural cause, it can have a genetic, infectious, metabolic, or immune cause. But actually, by far the most important cause, and what causes most people to have epilepsy, is an abnormality in their genes. So the most epilepsy is genetic. So just to remind you a little bit about some basic genetics. So you are all made of about 30 trillion cells, and each of those cells, apart from your red blood cells, have got genetic material in the nucleus. We call that the genome. That genome is made up of 23 pairs of chromosomes. And each of those chromosomes is a very, very, very tightly wound up DNA. And DNA is made of base pairs. There are 6 billion base pairs, and you can see at the end of this diagram here, each of these letters C, T, A, G represent one of the 4 base pairs that make up your DNA code. And a length of base pairs, or DNA, is referred to as a gene. So we've got about 20,000 genes in our genome. And what those genes do is they code for proteins, and proteins are incredibly important. They not only are important in terms of structure of your body, but they're also important in terms of function. So proteins, lots of proteins make cells and are important in the function of those cells. And then cells form together to make organs. And of course, you are a group of organs. Now, of course, remember, you all start off as a single cell. And now you've got 30 trillion cells. And so that means those cells have to be replicated a lot of times. And every single one of those cells has got the same DNA in it. And so that means your DNA has to be copied over and over and over. And it's actually very remarkable that that your body doesn't make more mistakes. It's incredible that we can copy our DNA so many times and we do it exactly perfectly. But, and how it happens is that the DNA, which is so these tightly bound strands, double strands, it separates, and then the body makes an identical copy, and one copy goes to one cell and another copy goes to another cell. And that usually happens perfectly, but every once in a while a mistake is made. And what that means is that you end up with one strand which is normal and the other strand which has got a change in the code. Now, some of those changes in the code are fine. It doesn't seem to impact you at all. In fact, we're all got lots of, I'm not identical to any of you. My DNA is completely different. So the code's always going to be different. And so some of the changes just make you what you are, or sort of a normal sort of things, or make no difference. But sometimes those changes can be what we call pathogenic variants. And so that's a change in the DNA code, which basically results in an abnormally formed protein, which then can't do what it's supposed to do. And that leads to disease. And in our case, we're talking about epilepsy. Now, here's a family, and if you come from one of these families where the green people have epilepsy and the gray people don't, you don't need a fancy professor of pediatric neurology to come along and tell you, I think it's genetic, because you know that it runs in your family. And also, even if you don't have a lot of family members, but you get 2 children, that seems very genetic. So people are pretty much find that easy to understand. However, most people who have epilepsy don't have a lot of people in their family who have epilepsy. Usually the situation is you just have a single individual, or you might have another affected individual who's really, great aunt so and so. So how is that possible? I'm telling you it's genetic, but it doesn't look like it's inherited. And so people often get confused between genetic and inherited. So not all genetic disorders are inherited. And so how does that happen? So why is nobody else in that family got epilepsy if it's genetic? Well, there's three reasons that could be. One is it can be what we call de novo. The other is it's due to multiple genes, and then it can be possible that the parent is mosaic. So I'll take you through those three concepts. So first of all, let's just look at a de novo variant. And so here we have a family, and each of these double ticks represents a single gene. And remember, everybody's got 2 copies of every gene. And so, and if you are blue, that means you have epilepsy. So what you can see in this situation is mom and dad have got 2 normal copies of this gene, and most of the siblings do. But this young lady has a 1 abnormal copy and 1 normal copy. So it's de novo because it's only in her. She hasn't inherited this from her parents. So how does that happen? Well, it happens really early on. So after the sperm and the egg get together and form that first cell, very, very early on, within the first few divisions, a change is made that causes what we call a pathogenic variant. And then that carries on for all of the replications and is in all of that person's cell, particularly in their brain cells. And if it's a variant that causes epilepsy, then it's a de novo variant. So that's what happens. So de novo variants are very common in terms of the really severe epilepsy. So a lot of the severe childhood epilepsies are caused by de novo variants. But the more common epilepsies we think are probably due to multiple genes being involved or polygenic inheritance. And so here, again, we have one individual blue who has epilepsy, but now we've got 3 genes. We've got a purple gene and an orange gene and a green gene. And each of those genes has got 2 copies. And so dad here has got 1 abnormal copy in his purple gene, and mom has got an abnormal copy in her orange and her green gene, but that's okay. Those variations don't cause you to have epilepsy. But unfortunately, dad's given his bad copy to his daughter and mom of the purple gene, and mom's given her bad copy of the orange and the green gene to her daughter. And that combination of having all of those abnormal genes has led this person to have epilepsy when the parents have not had epilepsy. So it's multiple abnormalities coming together to make somebody to have epilepsy. And then finally, it's possible that a parent may be mosaic. And so what we mean by mosaic is that, so it's a similar situation to the de novo, except for that one of the parents, and in this case, the mother, has 2 sorts of cells in her body. Some of her cells have got 2 normal copies, and some of her cells have got a normal copy and an abnormal copy. And she's handed on an abnormal copy to her daughter. So we'll talk about that in a bit more. And so that's what we call mosaicism, when you've got a mixture of cells in your body. And so how does that happen? So again, we start back here with our sperm and our egg. We have our single cell, and then it starts to replicate. And a mosaic situation happens when, after some cells have formed, one of the cells so later, so not right at the very beginning, after some cells have replicated, one of those cells has a copy error. And so that cell keeps copying that error into its cells. So those are the orange ones, but all the normal cells just carry on with what is normal. And so you end up with a person who has both some abnormal cells and normal cells. Now, if in epilepsy, if you don't have those abnormal cells in your brain, you're not going to have epilepsy. But if you have those abnormal cells in your reproductive system, so your eggs or your sperm, then you can still pass that on to your child. And so some research that we did that was published in the New England Journal of Medicine a few years ago was looking at how often this happens. And so what we did, and this is Heather Method's lab at, she was actually in Seattle then, and with Ingrid Scheffer in Melbourne, is we looked at around 200 families with severe developmental and epileptic encephalopathies, where their clinical or research testing had suggested that the child had a de novo mutation. So the blood test, the clinical blood test or the research blood test, when we looked at the parents, all we found and all the clinical tests found was 2 normal copies of the gene, but the child had 1 abnormal copy. And what Heather did is she did some really what we call deep sequencing. And what that means is she basically looked really hard at the DNA in the blood. And what she was able to look at when she had a really high depth sequencing was that there was a small number of cells that actually had an abnormal copy. And we found that that was the case in about 8% of individuals who thought on clinical testing that their child had a de novo variant. And that's important because, of course, if mom has, in this case, if mom, could be dad, but if one of your parents is mosaic, that means, sorry, if they're no mosaic, then you can't have another child because the abnormalities just occurred in the child. But if you're mosaic, then you have an increased risk of passing this abnormality on to another child. So why does it matter? Why is gene discovery important? Why am I doing this? And there's lots of reasons. And they fall into both clinical utility and personal utility. So first, let's look at clinical utility. So here's a figure outlining some of the importance of having a genetic diagnosis. So here's our little baby here who's got a severe epilepsy in childhood. And what happens to these kids when they first present? They're having lots and lots of seizures and their development's going nowhere. And it's a really horrible situation. Everybody wants to try to figure out why. So they get lots of tests and not just expensive tests, but invasive tests, including biopsies, etc. So if you can get in early and make a genetic diagnosis, then that means they don't have to have any more of those tests. So it allows the child to stop that diagnostic odyssey, we call it, and it allows everybody to stop focusing on the why and move on to think about what are we going to do now. It also, once you identified what the genetic disorder is, it allows people to have a much better knowledge in terms of what's going to happen. So better prognostic counseling. So we're able to then say, okay, if this is what's causing this child's epilepsy, this is what's likely to happen to them over time in terms of their development and what other comorbidities they may have. Do they going to need extra help in school? Are they going to need extra help when they're adults? It also, and this isn't just in the developmental and epileptic encephalopathies, but a fantastic study that was done in Scotland where every child under the age of 2 who had epilepsy was given genetic tests. That doesn't happen in New Zealand. It doesn't happen in many places, actually. It was part of a research project. And what they found is that 76% of these kids, and that's not just the severe ones, but also the children who had relatively mild epilepsy, having a genetic diagnosis, and 76% directed therapy. Now, and that's known therapy. So what that means is that it helped, it gave a better idea of which of the known anti-seizure medications that we had would be best for that child. So it directed better therapy earlier on. And then, of course, once we understand what the gene is, that helps us figure out what's that gene do? So how can we impact on the specific underlying, what we call pathophysiology of that genetic disorder? And that allows us to look for things like drugs that we already have available. So maybe not being used as anti-seizure medicines, but maybe being used for something else like cardiac diseases or other diseases, and so they're already available, but they're not being used in epilepsy. But because we understand what the underlying pathophysiology is, some of these drugs can be repurposed and start to use in epilepsy and can help these kids. And then also it helps in terms of developing novel therapies. So we're now talking about therapies directed directly at the genetic abnormality. And there's some fantastic stuff about to happen with the first ASO being released probably in the next couple of years. For one of the severe epilepsies of childhood. And then the other really important thing is, of course, it helps with reproductive counseling. So it allows people to have an informed choice in terms of what they might want to do with their family planning. So the other important aspect of having a genetic diagnosis is personal utility. And so Jenny Jeffries is a psychologist who did a doctorate with me. And so last year she published her work, and that was looking at the developmental and epileptic encephalopathy. She did some deep interviews and looked for themes from 18 families that had been given a genetic diagnosis for their developmental and epileptic encephalopathy from our research project, to see what that diagnosis meant to the family. So not the clinical aspect, but how it helped them. And in addition to that clinical aspect. And what she found was that, and what you've got to understand is a lot of these, because these kids are really severely ill and are having a lot of seizures, life's tough. And so these families have a high risk for relationship issues, mental health, and a lot of dysfunction. And they often are really focused on why has this happened to their child. So one of the things that it's really useful for is it gives people a sense of relief that they know that they didn't do anything wrong in the pregnancy that caused this, and they can then start focusing more on on moving on with how they can deal with the comorbidities and things that's likely to happen to their child. It also gives people a lot of knowledge, and knowledge is really useful in terms of the more you understand about something, the more you can get a sense, it helps you feel a sense of control. So that's really helpful in terms of mental health. And it also allows them to be able to hook up with other people who have similar genetic disorders. And they're, particularly in the developmental and epileptic encephalopathies, each of these individual genes, they form family groups who are fantastic in terms of supporting other families. And these are initiatives that are happening on Facebook and various other social media platforms. And so that all families from all over the world get together. And not only do they support each other, but they also fund for research to help get more research done on their gene. And then that also allows the families to have a sense of hope that maybe there is a new treatment available, or if there isn't, at least they know that somebody's really trying hard to find it. So there's a lot. So families really, you can't underestimate, on top of the impact on sort of treatment, how helpful this is for families with these severe disorders. So what have we found in terms of our gene discovery? Well, we've actually, with our collaborators that I work with around the world, we've been highly successful in identifying new epilepsy genes and showing the epilepsy syndromes that have been associated with these. But to you, these genes all just look like a bunch of letters on a page. And so, if it's your gene, it's really important. But if you don't know much about the gene, I always think people just think it looks like a jumble of letters. So I'm just going to talk about one gene we recently just published, looking particularly at the epilepsy syndrome associated with it. And that is a gene called PIGN, or as we like to call it, PIGN. And this was just published recently in Epilepsia. And so this was a gene that we identified in a family in New Zealand. And then we found, with our collaborators in Australia, we found some more children with abnormalities in this gene. And then we hooked up with a group in Denmark who was also collecting patients. And then through a research platform called Gene Matcher, we were able to find other individual scientists who had individuals with this genetic disorder. And we identified 26 individuals with abnormalities in the PIGN gene, and described in detail what actually happened, so what the phenotype, we call the phenotype in these individuals. And they all had a range of seizure types and a range of epilepsy types, but the vast majority of them had severe developmental and epileptic encephalopathy, with some of them having intellectual disability and epilepsy. And they all had moderate to profound intellectual disability, with not very many of them being able to walk or speak. And about half of them having a movement disorder. And this is a recessive disorder. So most of the disorder I've been talking about before we call dominant. And so this is a de novo dominant mutation. And so what that means is that the individual here has a disease. They only have to have one abnormal copy to end up with epilepsy. So that's a dominant condition. But PIGN is a recessive disorder. And so what that means is you actually have to have two abnormal copies to get the epilepsy. So the parents each have a single abnormality, and one good gene and one good copy and one abnormal copy, and that doesn't cause epilepsy. But in some of their children who unfortunately inherit both of their abnormal copies, they end up with epilepsy, and that's what we call recessive disorder. This is a picture of the protein that PIGN codes for, and each of these arcs represents an individual or a group of individuals, and it's so each of these points is an abnormality on this gene. And what you can see is this place here at leucine 311 tryptamine is a hotspot. So lots of these individuals, 10 of them, had abnormalities at this particular place. And one of the interesting things that we found in this gene was that if you had one of your abnormal copies was in that place, you were much more likely to have generalized seizures than focal seizures. And so that's an interesting finding and may have implications later on in terms of management for them. So that's pretty much all I've got time to talk to you today about. So I'd just like to acknowledge all the funders of our research. We're very grateful for the Neurological Foundation. Most of our genetic research is funded by the HRC and Cure Kids, and Freemasons Scholarship funded SHAMA. And we've been happy to have a philanthropic donation that comes directly to us. So thank you very much for listening, and I'm happy to answer any questions.